Hey, I wanted to jump on and create a really quick video. So what I have in front of me is an example of a schwannoma dataset. And here they've applied various drugs and genetic perturbations. And we can do things like differential expression and pathway analysis, and then ultimately use this data to create target lists and prioritize those targets. But let me show you how I actually got to this point on Pluto. So when you are ready and you have your FASTQ files, you can start a new experiment. So we will call this just RNA-seq schwannoma, and then you can select from any type of experiment, bulk RNA-seq, single cell, proteomics, and ATAC-seq as well. But all of these data types are available in Pluto, and they have pipelines associated with them. Then you can select your organism. So these can also be configured to your preferences. I'm going to select human. And then from here, this might be the point where you normally send off your FASTQ files to a consultancy to analyze, but in Pluto, you will just go ahead and choose those FASTQ files and upload them directly here. And when those are done uploading, you can just click I'm done uploading files. And then we will select our sample metadata file. And this is something you might start off the platform in Excel and just create all of the variables that you want to track in that file, but you can always edit it as much as you need to in the platform. So we will save and continue. And then from here, we'll be prompted to just fill out a few parameters to execute this pipeline. So in this case, it's genome, library type, and strandedness. The pipeline that we use is the NF-core RNA-seq pipeline. That code and all of its methods and settings are available to you, so that is fully transparent. And then from here, we can just click Run Pipeline, and that is going to execute the pipeline for you. So you're in full control of that, and that typically will take a few hours, and then you will get your results delivered to you. And you can track this pipeline, see that it's in progress, and then you can always see exactly what parameters were set. So obviously this might take an hour or two to complete, but when it's done, and I'm going to do this kind of cooking show style, you'll have this nice experiment view. So you can track notes about the experiment, and down below, every pipeline will deliver a multi-QC report. So you can see things like sequencing quality and duplication and so on and so forth. When you're ready to actually begin your analysis, you can head over to this Analysis tab and click on Canvas. And then that will take you to this nice kind of scientist's whiteboard where you can actually start running analyses and creating scientific stories. So you can see a few of the analyses that I've already generated, but I'll just walk through how one of your scientists might do this. And this is entirely through non-coding graphical modules. So I'm going to click Create New Analysis and select differential expression. And then any of the variables that I included in my sample metadata, which is down below here, are available to my disposal to analyze. So for example, if I want to look specifically at treatment and gene perturbation, I can actually kind of stack those two variables, and I will select bergatinib control, or sorry, I'll do bergatinib knockdown as my experimental group and bergatinib control as my control group. And then I can click on Run Analysis. This will kick off a bioinformatics script in the cloud that might take 10 seconds to a minute to run, just depending on size of the data. And then once that visual has been created, you can go in and make any adjustment that you need. So for example, we could add a p-value threshold line. We could go in and change colors and make this, say, pink, for example. And then we can also label genes of interest or create target lists here. So for example, if I want to label a public list of genes for E2F targets, I could do that and save changes. And then all of those genes will get automatically labeled for me. I can also lasso the top differentially expressed genes here and create a whole new list from that. So you'll see this kind of highlighted, all of those genes. I can copy those and save them to a list. So let's say I want to call this bergatinib target selection results, and then save those genes to a list. And then if I go over to target management in Pluto, I'll see that that list has now been created. And from here, I can actually start to rank and prioritize and add evidence to these targets. You can also do things like gene set enrichment and summary plots. You can also create concordance analysis, so that's where you actually start to compare across different experiments and look at targets that appear in your RNA-seq data and your single-cell data and your proteomics data. So lots of really useful tools for multi-omics integration as well. If you have any questions or this generates any questions for you, please let me know, and I hope we chat soon. Bye.